Well, hello everybody, Mike Prevost from MikePrevost.com here, and today I want to talk to you a little bit about lipoproteins. Lipoprotein is a particle that carries lipid in the blood. It's a protein particle that carries lipids, and in this case, we're talking about triglycerides and cholesterol. We're going to talk about metabolism of lipoproteins, and we're going to talk about endogenous and exogenous pathways. Endogenous is inside of the body, so we're talking about the body's production of those lipoproteins. And exogenous is external, so we're talking about actually eating or consuming lipids and how they're packaged and metabolized. Okay, let's get started. Okay, I want to start off talking about the exogenous pathway. Okay, so in this case, we're consuming lipids, so we're consuming cholesterol or fats, which are basically triglycerides. How does the body package them up, and what does it do in terms of metabolism? Well, we digest all of those lipids and we absorb them in the small intestine. And the cells of the small intestine package them up into something called a chylomicron. And a chylomicron is basically a protein shell consisting primarily of a ApoB48 protein that's packed with those lipids. So we basically put those lipids, triglycerides, cholesterol, and phospholipids, into that ApoB48 shell. It becomes something called a chylomicron, and that's pushed out into our lymphatic system, and eventually it enters the circulation. Now, when that chylomicron enters the circulation, our HDL particles, and you might recognize HDL as high density lipoprotein, or the so-called good cholesterol, right? There's a HDL particle, and it gives some proteins to the chylomicron. Basically C1, C2, C3, and ApoE. It gives those proteins over to the chylomicron, and now the chylomicron is a mature chylomicron. It has all of those apoproteins associated with it. Now those apoproteins that it gets, they do specific things, and so I've got a little chart here that tells you specifically what those apoproteins do. And we're not going to get into too much detail on all of these, but for some of you who want the detail, ApoC1 activates LCAT. That's the cholesterol acyltransferase protein. We're not going to talk too much about that, but it esterifies cholesterol. ApoC2 we're going to talk about in a minute. It activates tissue lipoprotein lipase. C3 inhibits C2, so it inhibits the action of C2. And ApoE is used for recycling the chylomicron. Okay, so let's talk about what happens. We got this mature chylomicron here. This mature chylomicron now has all of those proteins associated with it that it got from the HDL, and it circulates around and it counters tissues of the body, like for example a fat cell or an adipocyte. On the tissues of the body, on the surface of their cell membrane, they have this thing called lipoprotein lipase. Okay. That lipoprotein lipase, it interacts with the ApoC2 on the chylomicron, and it triggers the transfer of lipids from the chylomicron to the tissue. That's what lipoprotein lipase does. It's a lipase. It breaks down those lipids and it transfers them into the adipocyte. Okay, so that's how the chylomicron delivers the lipids that you've ingested from the small intestine out to the tissues of the body. So it packs it up into the chylomicron. Chylomicron gets a little help from the HDL, wanders over to the tissues, and then starts delivering those lipids to the tissues. That's what a chylomicron does. Now, once it's dumped out all of its lipids, it becomes a chylomicron remnant. And in that case, the chylomicron remnant is recycled primarily by the liver. And the liver will take up the chylomicron remnant through the ApoE, through what's called an LRP receptor. Some of them are taken up by the LDL receptor, but very few. And so now the chylomicron is taken up by the liver. It's broken up and recycled. Any lipids that are remaining are pushed out as bile, and the proteins are recycled by the liver. Now, one thing to note about these chylomicrons is the half-life of chylomicrons is really, really short. So they are full of triglycerides. So you might think if we're measuring blood triglyceride levels in a lipid panel that we're measuring chylomicrons, but we're not measuring chylomicrons at all. Really, within a couple of hours, chylomicron levels would be very, very low. And if you do an overnight fast, your chylomicron levels would be expected to be almost non-existent. So when we normally measure blood triglycerides with an overnight fast, we're not measuring these triglycerides. We're not measuring endogenous triglycerides, or triglycerides coming from the small intestine from the food that we've eaten. And we'll talk about what we're actually measuring in a bit. So that was the exogenous pathway, and that was metabolism of lipids that we consume or eat. But the body produces a lot of lipids, primarily in the liver, and that's the endogenous pathway. So let's talk about the endogenous pathway. And it all starts with the liver. The liver makes something that's sort of similar to a chylomicron, but it's got different proteins associated with it. It's called the VLDL, a very low density lipoprotein is what it stands for. It's a protein shell consisting of an ApoB100 protein, and it's packed primarily with triglycerides. But it also has some cholesterol and some phospholipids, but it's mostly a triglyceride particle, this VLDL. And it's the way the liver exports triglycerides. So if the liver is manufacturing a lot of fat, it gets rid of that fat by exporting it as a VLDL. Fat in the liver is basically toxic, especially when it builds up. You've probably heard of a fatty liver. Cirrhosis is a fatty liver, or non-alcoholic fatty liver disease, which can happen in diabetes, is a fatty liver. And so in those conditions, the liver would export a lot of that fat. It would be making lots of VLDL to get the fat out of the liver. Okay, and that fat's exported as triglycerides in a VLDL particle. Okay. And like I said, it also contains some cholesterol and phospholipids. Now that VLDL, it's going to encounter an HDL, just like the chylomicron did. And the HDL is going to do the same thing it did for the chylomicron. It's going to transfer over some apoproteins, all the same ones that you saw earlier. And those apoproteins are going to associate with that VLDL. And that's what makes it a mature VLDL. Now that VLDL can go out to the body tissues. And just like the chylomicron, this should look really, really similar. It uses the ApoC2 to interact with lipoprotein lipase to give triglycerides to the tissues. So you can see what's happening in step one. The liver packages a VLDL with cholesterol and a lot of triglycerides. The VLDL matures in step two. In step three, just like the chylomicron, those triglycerides that it got packaged up in the liver are now given to body tissues through the lipoprotein lipase. Now when that happens, after this VLDL gives up some of its triglycerides, now it's a little bit more concentrated in cholesterol now since it's given up some of its triglycerides. When it does that, it becomes what's called an IDL, or an intermediate density lipoprotein, because triglycerides are very low density. When it gives up some of its low density stuff, it becomes a little bit more dense. That's how we make an intermediate density lipoprotein, or an IDL. Now, one thing about one of these apolipoproteins, apoprotein C3, we mentioned earlier, interferes with the function of C2. So it would interfere with this process of transferring lipids from the VLDL to the body tissues, in this case an adipocyte. So if you have a high level of C3, you tend to have these lipoprotein particles that are stuffed full of lipids and have a hard time transferring those lipids to body tissues. And that tends to be atherogenic. Atherogenic meaning promotes atherosclerosis, or heart disease. So a high concentration of ApoC3 is not a good thing. Production of ApoC3 can be increased by a really high carbohydrate diet, especially a high sugar diet, and also by inflammation, which is one of the ways those two conditions can contribute to heart disease. Okay, so now we've kind of traced a VLDL all the way to producing an IDL, or an intermediate density lipoprotein. So now let's talk about what happens to that IDL. So we've produced that IDL from a VLDL. Now this IDL has sort of two possible fates. So one fate of that IDL is it can go right to the liver and through the LDL receptor, it can be absorbed. And once it's absorbed, it's typically broken up by the liver. The lipids inside of the IDL are pushed out as bile. The proteins are recycled. So one fate of the IDL is just to get taken up by the liver and recycled. And that happens through the LDL receptor. About 50% of the IDLs get absorbed, and it varies from person to person. But some of the IDLs, instead of being absorbed, they interact with hepatic lipase. And hepatic lipase is very similar to lipoprotein lipase. In this case, the IDL, again, it gives up some of its triglycerides, just like a lipoprotein lipase, except for this time it's given the triglycerides to the liver. And then that IDL, after it gives up some of its triglycerides, becomes an LDL, or a low density lipoprotein. Now you might recognize this one. You've probably heard of HDL cholesterol and LDL cholesterol. Well, here is the LDL cholesterol. We've had a particle now that started out as a VLDL that was high in triglycerides and relatively low in cholesterol, but it kept dumping out triglycerides, right? It dumped out some here as a VLDL, and then it became an IDL, and then it dumped out some more here at hepatic lipase, and now it's triglyceride poor, but it's cholesterol rich. It's relatively more rich in cholesterol now because it got rid of a lot of triglycerides. And so when we say LDL cholesterol, what we're talking about is the amount of cholesterol that's carried in these LDL particles. So this is the so-called bad cholesterol. So what happens to this LDL particle, this LDL triglyceride particle? Well, a couple things can happen. One thing can happen is this LDL can interact with HDL again. And when it interacts with HDL, it can, through a little handshake agreement, swap out some lipids. So basically the LDL encounters the HDL and says, hey HDL, I'll give you some triglycerides if you give me some cholesterol. And that happens through an enzyme called CETP, cholesterol ester transferase protein. So it encounters an HDL in the bloodstream, and through CETP, this LDL gets even more cholesterol, and it gives away even more triglycerides, and so it becomes more dense and more cholesterol rich through its interaction with HDL. Now notice the HDL is giving up cholesterol to LDL. That'll be important later when we talk about reverse cholesterol transfer, because we'll talk about where the HDL is getting that cholesterol from. So now we have this LDL that's become pretty cholesterol rich, and it stays in the circulation, and when it's in the circulation, something can happen to that LDL. In fact, the longer it stays in the circulation, the more likely this is to happen. And what can happen is some of the lipids on that LDL can become oxidized, and then we have an oxidized LDL, and that's not a very good thing, and we'll talk about why oxidation of the LDL is really bad in a minute. So now we have some LDL and we have some oxidized LDL. So let's talk about what can happen with this LDL and oxidized LDL. What I didn't mention in the last slide is that most of the LDL that's produced gets recycled in the liver right away. About 80% of it, not right away, but pretty quickly. So about 80% of it gets recycled in the liver. But some of it, a smaller percentage, is going to be taken up by macrophages. Macrophages are immune system cells that are all over the body, and one place where they're located is basically inside of the walls of the vessels, the blood vessels. And so those macrophages can actually take up that LDL through the LDL receptor. So that's one fate of LDL. In addition to being recycled by the liver, taken out of circulation by the liver, it can be picked up by the macrophage and taken into the macrophage by the LDL receptor. Well, we also have oxidized LDL. So what's the big deal with oxidized LDL? Well, it turns out the LDL receptor here is a regulated receptor. That means when the macrophage gets full of cholesterol, the LDL receptor goes away and it no longer takes up much cholesterol. And so through the LDL receptor, you can't get a really big fat macrophage that's just stuffed with cholesterol, what we call a foam cell. You can't make a foam cell through the LDL receptor. But if you have oxidized LDL around, well, there's a problem because there's two other receptors, the SRA receptor and the CD36 receptor, which are not regulated, but they only take up oxidized LDL. So if you have a lot of oxidized LDL around, those receptors will just gorge on oxidized LDL. It's like a buffet, and they don't know when to stop eating because they're not regulated. As long as there's oxidized LDL around, they'll continue to take it up, and they will stuff that macrophage beyond full. Now, when the macrophage starts to become really, really full of cholesterol, it becomes a foam cell. And a foam cell is one of the early stages of atherosclerosis or heart disease or blockage of arteries. You get fatty streaks, which are basically foam cells. And so it takes oxidation of the LDL to really produce a foam cell and cause atherosclerosis. The normal LDL through the LDL receptor will not form a foam cell because it's regulated. So that was the endogenous pathway for LDL metabolism, or low density lipoprotein metabolism. So this is the whole picture, put together from soup to nuts. So let's just do a really quick run through again. Okay, so the liver packages up lipids, triglycerides, cholesterol, and phospholipids in a VLDL, which is immature. Now the VLDL interacts with the HDL and it gets some of these apoproteins and becomes a mature VLDL. And now that C2 apoprotein allows the VLDL to interact with lipoprotein lipase and give it some triglycerides. And once it gives up some triglycerides, it becomes an IDL. Now, most of that IDL is absorbed in the liver. I'm sorry, about half of it is taken up into the liver for recycling. But about half of that IDL interacts with hepatic lipase, gives up more triglycerides, and becomes an LDL, right? Now, that LDL can interact with HDL, and it can take some cholesterol from the HDL and give away more triglycerides, and it can become a more dense LDL, and it can also be oxidized, okay? But most of the LDL really is recycled through the liver. A small percentage of it, though, is taken up by the LDL receptor, and the oxidized LDL is really taken up greedily by the macrophages. And if it takes up enough oxidized LDL, it becomes a foam cell, and the foam cell causes fatty streaks in the arteries, and they also cause the release of lots of inflammatory cytokines which drive atherosclerosis. So that's the process whereby LDL can cause atherosclerosis. Well, that was LDL, the so-called bad cholesterol, and we ended that one on a bad note with atherosclerosis, right? So I want to talk about HDL, which is often called the good cholesterol, and we'll talk about the endogenous pathway of HDL metabolism. So HDL is basically a lipoprotein particle, just like the LDL. Instead of having an ApoB100 associated with it, it has a different protein associated with it. It has an ApoA1. And HDL is made in really two tissue compartments of the body. About half of it is made in the small intestine and about half in the liver. And what happens is those tissues basically secrete a lipid-poor ApoA1. ApoA1 is basically the shell that's going to contain the cholesterol and the triglycerides. But then as soon as that ApoA1 is released by the small intestine or the liver, it interacts with something called an ABCA1, which immediately gives some lipid to the ApoA1 shell. So, for example, the small intestine secretes a lipid-poor ApoA1. That ApoA1 immediately interacts with ABCA1 right there on the small intestine, and it gets some lipids. And it becomes an immature HDL. Okay. Now, that HDL, immature HDL, can be further lipidated. It can pick up some more lipids through something we talked about just a minute ago, CETP. Right? And we showed HDL interacting with LDL through CETP. It can also interact with VLDL and other particles, but what would happen is that immature HDL through CETP would pick up some more triglycerides. It'd become more lipidated, and then now, we have a fully functional HDL. It's got some cholesterol and some triglycerides in it. Okay. Now those lipids initially go into the outer membrane of the HDL, and they're moved to basically to the inside of the HDL through an enzyme called LCAT. So it's taken unesterified cholesterol and it's moving it to the membrane of the HDL, and then the LCAT esterifies the cholesterol and moves it internally. Just a small detail for those who are into the small details. So now we have a mature HDL. Well, what happens to the HDL? Well, lots of things can happen to the HDL. One of the things is that the HDL now can go out to the body tissues and it can take up lipids. So our HDL can go here, it can interact with body tissues and macrophages, and it can take cholesterol from those body tissues. So our macrophage that was gorging itself on cholesterol and trying to become a foam cell, our HDL can come along and say, hey, look, you've had too much cholesterol, I'm going to take some of that from you through ABCA1. And that's called reverse cholesterol transfer. So our LDLs deposited the cholesterol in the macrophage. The HDL comes along and says, hey, I don't want you to become a foam cell. I'm going to take some of that from you. And so it takes up some of that cholesterol. And the HDL can do the same thing with some body tissues, other body tissues. It can take up some cholesterol. Now, what does the HDL do with that cholesterol? Well, one of the things it can do is it can interact with a receptor called SRB1 on the liver, and it can just give that cholesterol to the liver via SRB1. So follow the flow here. So we have an HDL which is going to our macrophage. It's pulling cholesterol out of the macrophage, and then it's giving it to the liver. And then the liver can convert the cholesterol to bile and excrete it out of the body. So that's reverse cholesterol transfer. We can basically take cholesterol out of our vessels and give it back to the liver. And that's a good thing. Well, the other thing that can happen to the HDL is it can give that cholesterol to the LDL, which we showed in the last slide, and then that LDL can be taken up and recycled in the liver, and the cholesterol in that LDL can be excreted as bile. So that's another way that the HDL can take cholesterol out of our vessels, out of the macrophage, and prevent it from becoming a foam cell and hand it off to the liver to be excreted. It can do that through the LDL or through the SRB1. Okay? And really it can do it through the VLDL as well, which is what we're depicting here. And finally, when the LDL is done, when it interacts with the liver, it can be broken up. It can be internalized through an unidentified receptor. There's a receptor on the liver that takes it up and internalizes it that's not been well characterized yet. It can interact with this SRB1 and then just return to circulation and not be taken up by the liver. Or it can be taken up by the liver and then recirculated again. So it can either be taken up by the liver and broken up, this HDL. It can be taken up by the liver and then re-excreted to be recirculated. Or it can just hand off some of the lipids to the liver and then go right back into the circulation. Those are the three fates of the HDL. So one more concept to wrap up with, and this has to do with exogenous metabolism of cholesterol specifically. And what I'm talking about is cholesterol that you eat. So let's talk about how that happens, because this has an important implication. So when we consume cholesterol, the enterocyte, which is basically the cells that line the small intestine, they take up cholesterol, non-esterified cholesterol, and plant sterols through this receptor called NPC1L1. Okay, that's the Niemann-Pick C1 lipoprotein. Strange name, I didn't name it. That's just what they call it. Okay. Now, an important thing to know: the NPC1L1 only takes up non-esterified cholesterol, and most of the cholesterol that you eat is esterified. Now, we have an enzyme in the small intestine that can de-esterify that cholesterol, but it doesn't get all of it. And so the first point is that a lot of the cholesterol we eat is not absorbed at all because it's esterified, and it does not all get unesterified by the enzymes in our small intestine. So some of it just flows right through. But the cholesterol that's unesterified and plant sterols, they get taken up by this NPC1L1 protein. Okay. Now, once they're taken up, the non-esterified cholesterol and plant sterols, some of the cholesterol gets esterified, some of it doesn't, and none of the plant sterols get esterified. So all of the cholesterol that does not get esterified and all of the plant sterols are then carried back by the NPC1L1 to this ABCG5G8. It's called an ATP binding cassette protein, which basically pushes it back out. Okay, so boy, that sounds complicated, but it's really not too complex. Basically, the NPC1L1 takes in the non-esterified cholesterol and plant sterols, and then the ATP binding cassette pushes back out all of the plant sterols, because we don't really have a use for that, or almost all of the plant sterols, and any cholesterol that doesn't get esterified. Okay, so the little bit of cholesterol that does get esterified that we've absorbed gets packaged into a chylomicron and pushed out into the lymph. We've talked about that already. So why is this important? Well, really 2 points. One is, we've already said that some of the cholesterol remains esterified, and so it just passes right through and we excrete it. The other point is that this process, this loop right here, is regulated, and when body cholesterol levels, blood cholesterol levels are high, less cholesterol gets esterified here and more gets pushed back out, and so it's regulated. And so what does that mean? That means that it's really hard to drive blood cholesterol up by eating cholesterol, because once your blood cholesterol level gets up, your enterocyte's just going to excrete more. And so there's not a strong connection between cholesterol you eat and blood cholesterol levels. And it's taken us a long time to learn that. 20 years ago, it was thought that consumed cholesterol you eat in the food, becomes blood cholesterol. We need to eat less cholesterol, and that's really not believed anymore. And those recommendations are really going away. The recommendations to limit ingested cholesterol, because of this whole process that, ingested cholesterol, it's very well regulated, and when blood cholesterol levels are high, you just don't absorb as much. And that's the exogenous pathway. So, so where does blood cholesterol come from? Why does it get high? Well, we talked about it a minute ago, and that's the endogenous pathway. That's the body producing cholesterol and packaging it up in VLDL particles. That's where blood cholesterol comes from. And I mentioned earlier in the lecture that when we measure blood triglycerides, we're not measuring those chylomicrons, because the chylomicrons have a short half-life. Really what we're measuring is the triglycerides in the VLDL and the LDL particles. That's what we're measuring, not the chylomicrons. And so that's not exogenous either. It's endogenous. So when we do blood lipid panel, really we're not measuring exogenous lipoproteins. We're measuring endogenous lipoproteins, or lipids that the body manufactures. Well, that's about it for today, folks. I know that was a real handful. I wanted to share this because it took me a long time to put together this simple little series of pathways. I had to read dozens of research articles and, little by little put the complete picture together to really understand the flow of all the lipoprotein particles. And those who are struggling like me to understand lipoprotein metabolism, I figured could benefit from the work that I did to understand all of the background. Anyway, I hope you found it useful. If you did, please spread the word. If you got questions, head on over to my site and send me an email or drop a note in the comments block, and spread the word. Thanks for listening.